So now that we have performed major of the analysis in our research paper, the last step is to validate the results. We can say to validate the proteins that we have included. So for that we need the protein structure. So the lecture is for that. We have to perform the protein structure prediction analysis. So we are going to make the structures of our protein. So first of all, protein 3D structure will be performed by using the iTaser online tool. It depends on you if you want to use iTaser. If you don't want to use iTaser, you can use another tool as well, which is the Swiss model, which is developed by the Swiss stock company. So the protein sequence was submitted to iTaser to obtain the 3D structure, but there is a drawback using iTaser. When you will use iTaser, you will get your result after 48 hours. First of that, if your protein is lengthy, not lengthy, it doesn't matter. You will get your result after 48 hours. I think so. But if you use Swiss model, you can get the results of the protein quickly. Or if you use AlphaFold, which uses the machine learning technologies for the protein structure prediction, you can get your result at the main task, at the main point, at any time there in 5 to 10 minutes. After performing the protein 3D structure, we will validate that proteins. To validate, we will use the save server to check if our protein are predicted correctly or not. So now if we move to our website, here we will see the server is completed prediction for 7,557,396 proteins. So we know that this takes a lot of time and things like that. So what I'm gonna show you is that how to use this iTeaser protein online server. So for that, what you need is you need your protein sequence. So we are just gonna go with an upregulatory gene, which is CCL20. So I'm just gonna paste the CCL20 protein residue in FASTA format. And after that, I'm gonna put my company's email and password there. So you cannot use your Gmail or public account. So you have to be assigned by any institute or your company to use that. So my company is Bioinformatics.pk, where I work, and I'm the CEO of that company. So we have created mails for us to use the email accounts for the protein structure predictions. So after that, I'm just gonna click on run iTaser. So when I'm gonna click on run iTaser, it will show me the protein has been successfully submitted to the iTaser server. Your job ID is this. You will be notified by the email for your protein. So let's head back to the iTaser queue. Here we will see the job is pending. So your protein name and bioinformatics.pk, it is submitted by that. So the protein, if you can see, we have a lot of other people that are currently modeling their proteins as well. So if we click here that I have modeled recently, so model 1 cannot be, so I will keep this file. So how the results will be shown? You will be shown like this. If we scroll down, I got another job which is here. So here is the protein structure of that model. So if I'm gonna click on model 1, I'm gonna get the results, which will be in PDB file, protein and structure file. So I'm gonna open this file and show you as an example. So this is how to use the iTaser server to obtain your PDB files. So there is another way as well to model your proteins. So here you can see the another model that we can use is our Swiss model database to use the modeling of your protein structure. So paste your target sequence here. So we will paste our target sequence here, and after that it will validate it. So then we will search for templates. Click on search for templates. Then it will search for template and move us to the queuing section. So the once it is done, it will start showing animations of the proteins that are running. So here we can see the protein is currently running against HBV lids, and then it is aligning. After aligning, the model will be built. So now let's just, while this is performing, we will move to the save server. How to use the save server to validate the protein structure. So after search for the save server on your favorite browser. After that, click on choose files and then choose your model and then run programs. Click on run programs. So while it is running, let's check on the Swiss model. So it is currently running, and after that, yes, we can see after that you can check for your protein model. So verify 3D, determine the compatibility of an atomic 3D with its amino acid sequence. Click on that, and then we will see, yes, the 32.79 residues have averaged 3D to 1D. So this fewer than 80% protein have scored less than 1. So this protein is not structurally validated. You can also check the sterilized chemical quality. If you have a bigger protein, that will be automatically saved. Yes, so I choose the small proteins for my structure analysis. So error, so you can check all the protein subsections that you want to. So the overall quality score of our protein is 79, which is not that bad. It is somewhere good. So ProCheck is giving us the result in less than a minute as well. So while that was happening, so we have the models here. When we search for the templates, the Swiss models gave us the template of our protein. So here we can see AlphaFold. AlphaFold has given us the method of this protein structure prediction. So it is a monomer, no ligand is attached to it. So I'm just gonna go with this and build the model. So now the model is being building. We can move to the save server, and out of 8 evaluations we have a 6 error in our proteins, but the quality scores are still good. So our structure is somewhat good. The IT server protein was somewhere good. So let's just close the save server and focus on our model that we built. So click on structure assessment, and then we can see the Ramachandran plot here. So here is the protein structure that we made, and you can see your Ramachandran plot here of the protein. That is a protein structure validation. And if we if we have seen our protein correctly, we can also save the protein. So you can download the coordinates, you can download the archive and all the things that is available here. So that is how to model your proteins and then validate your proteins. So that is how it is done, and then you can download the data by PDB in PDB format, which is here. So there is a 01 model, and here you can see here is the protein structure. So for visualization, we will to include these figures in our research paper, we will make this more appealing. For that, we will be using the software Chimera. You can download this. This is freely available tool. You can download this on your research on your browser and then install it onto your PC for visualization. So after that, we will make the protein somewhere more appealing to be in the research paper. So for that I'm gonna use the presets of the protein, and all after that I am going to make a figure out of this protein as well to include in my research area, in my research paper. So I'm gonna go with this, the white protein head, and after that I am just gonna move to my tool section, and after tool section I'm gonna go with the, I think I will be going with the flat ribbon. No, so I will choose this rounded ribbon section. So I think this looks quite appealing to be in the research paper, but to I want to make this figure like this, somewhere like this, so the protein clashes will be on. So if I move to atom and bonds, I can show this as well. And if we move to the focus of the protein, so here we can see, if we hide our atom and bond and show the surface of the protein, here we can see where the protein ligand will be docked as well. So I'm just gonna move to the ribbon and surface and hide this, and I am gonna focus the protein somewhere like this, so it has a better visualization, and then save the image. So we will save image dot png in our desktop. We will get the figure on our desktop. If we can move in here we got the figure of our protein. So we will include this figure in our research paper as well. So here we can see we have the figure. So you can model all the 2 proteins, all the 5 proteins that you want, but for showing you, I'm just gonna model one single protein and include it there. So that is the 5th analysis done. After that we have our last analysis, which will be the protein ligand and docking intersection. So for that we will be using the Swiss model tool online, which is also an online tool. So you can use another docking tools as well if you want to. I'm just gonna show you methodology of how to do that. So that is pretty much it for that, and after that lecture I'm gonna show you how to change all this into a proper research paper. So stay tuned for that one, and thank you so much.